Hello dear students, welcome to Genesis. Well, in this class I am going to explain the structure of nephron. These are also known as renal tubules, kidney tubules or uriniferous tubules. What are these? These are those structures which are present inside the kidney. That means the kidney is composed of many of these structures. Now there are almost 1 million nephrons per kidney. So let's quickly see the structure. Structure of one nephron looks like this. It is a tubular structure. So it starts with a cup-shaped part which is known as the Bowman's capsule. Then it goes into a convoluted part, and since this part is close to the Bowman's capsule, so it is in proximity. So we call this as proximal convoluted tubule. This then goes down to form a U-shaped structure known as the loop of Henle. The loop of Henle then comes up into another convoluted structure, but since it is distant from Bowman's capsule, we call it distal convoluted tubule. So this is proximal convoluted tubule and this is distal convoluted tubule, and the distal convoluted tubule then opens up into a duct which is known as the collecting duct. So if I have to make a flow chart to tell you how the tubular structure starts and ends, then it starts with Bowman's capsule, then PCT, then loop of Henle, DCT, and then finally the collecting duct. Now what exactly is happening in these structures? Remember one thing, the blood enters into the kidney through a pipeline which is known as renal artery, and by now you already know that the blood in the renal artery has oxygen, but it is impure because it is filled with nitrogenous waste. So the pipeline or the blood vessel of that renal artery that enters into this structure is known as afferent arteriole. So you might be confused between artery and arteriole. Go back to my video and please see that the artery branches into finer structures which are called arterioles. So basically the renal artery branches further into structures which are known as arterioles because they have to enter in these tubular structures. And there are millions of them in one kidney. So you can imagine the number of arterioles that are also there. So from the renal artery comes out this structure which is known as afferent arteriole. Always remember, any blood vessel that enters into a structure starts with A, afferent arteriole, and the one which comes out of the structure starts with E, which is efferent arteriole. So the blood carrying the nitrogenous waste will now enter into this cup. There it is going to throw out its nitrogenous waste. And how is it going to throw out the nitrogenous waste? This wall of the Bowman's capsule is very thin, so it is ideal for filtration. On top of this, the filtration here happens under very high pressure. That is why this filtration is known as ultrafiltration. Now, how is this pressure getting generated? Look carefully. The diameter of the blood vessel which is entering into the Bowman's capsule is thicker compared to the diameter of the blood vessel which is coming out of it. And here is the main magic. So basically the blood which is entering here will be under very high pressure because it can enter with speed, but while it is coming out, since the tubule is narrow, it faces that pressure, as a result of which there is huge pressure of blood that gets generated here that compels the nitrogenous waste to be thrown out of the blood and they start to get collected in the Bowman's capsule. Now there are two things which you have to keep in mind. What will happen to the blood and what will happen to the nitrogenous waste? So the nitrogenous waste is getting collected in the Bowman's capsule and it has that so-called flow from Bowman's capsule to proximal convoluted tubule to loop of Henle, then distal convoluted tubule, and then finally to the collecting duct. You must be wondering where this duct leads. All the ducts, all the collecting ducts of million nephrons that are present in one kidney will finally open up and pour their content into the ureter. If you remember, we have a pair of kidney, we have a pair of ureter which finally leads to the urinary bladder. So basically all of that urine is now going to be poured into the ureter, and from ureter it will go to the urinary bladder. So this part is one part. The second part is what happens to the blood. So the blood when it entered here was rich in oxygen and also had nitrogenous waste, but the blood that is coming out of it now has less oxygen but has no nitrogenous waste. So what should happen to this blood? This should go back to the heart. From the heart it will go to the lungs where it will be purified. That means oxygen is going to be added to it and then it will be circulated into the entire body. But how is that happening? If you look here, the blood vessel that comes out is known as the efferent arteriole, but this efferent arteriole does not have a very smooth journey. It goes and it again branches and forms a network all around the tubule. tubule. And you know what is this network called? It is called vasa recta. What is the function of this network? I will explain you in the next class, where I will explain urine formation. As of now, just understand there are going to be exchanges that will happen between the blood here and all the filtrate that is getting collected in this part. So there are going to be some exchanges which are going to happen here. So the urine is not yet formed. There will be some exchanges, and then whatever comes out is known as the urine. Now, what will happen to the blood vessel? The blood vessel which is coming out of the Bowman's capsule will form vasa recta, that means a network, and then it goes out and joins the renal vein. Obviously, it will join the renal vein because the blood vessel that comes out of the kidney is known as the renal vein. Now, one question comes to your mind: what will happen to the renal vein? The renal vein will go to the heart, but how will it go to the heart? It will open up into the vena cava. Which vena cava, superior or inferior? Since kidney is located in the lower side of the body, it will go and open up into the inferior vena cava, and then it will be circulated from the heart. It will go to lungs, purified, back to heart, and then circulated to the entire body. So this is how every single nephron inside your kidney is functioning. Do not get confused between the two structures that are overlapping here. One is this structure, which is the structure of the nephron. Another is this network of blood vessel, which is known as the vasa recta. If you are asked to write a flow chart showing the flow of nitrogenous waste, then you are just going to mention Bowman's capsule, PCT, loop of Henle, DCT, collecting duct, and this collecting duct finally open up into the ureter. That's all. But if you are asked to write the flow of blood, then it will be renal artery, then afferent arteriole, then efferent arteriole, which will open up into vasa recta, from here into the renal vein, and the renal vein will finally open up into inferior vena cava. One more part that you must know, this part where there is this blood vessels and the cup, the total conjugated portion of these two is known as Malpighian tubule. So I have drawn it separately here. So basically the cup containing the blood vessel, blood capillaries inside it, this entire structure is known as Malpighian tubule. Or a simpler way to understand this is Malpighian tubule is equal to glomerulus plus Bowman's capsule. So now you already know that the kidney is made up of many such nephrons. But when this kidney is seen from above, we find that the region of the kidney, which is close to the capsule known as the cortex, it appears granular, whereas the middle part of the kidney appears striped. Okay, why is this? Because if you look at the arrangement of the nephron inside the kidney, I've just drawn one nephron for our convenience, but you can imagine there are millions such nephrons which are arranged in this way. So the Bowman's capsule, the proximal convoluted tubule and the distal convoluted tubule, this part, it remains close to the cortex. That is why the cortex appears granular. So there are three structures of a nephron that are present in the cortex. One is the Bowman's capsule, second is the PCT, and third distal convoluted tubule. Whereas the loop of Henle and the collecting duct that lies in the medulla of the kidney, and that is why it has a striped appearance. So there are two structures in the medulla. One is the loop of Henle, another is collecting duct, because of which the kidney shows a striped appearance in the medulla. And there are three structures which are your Bowman's capsule, PCT and DCT in the cortex, because of which the cortex looks granular. Now, as you already know what is ultrafiltration, let us understand what all happens inside this. So the blood, when enters into the Bowman's capsule, under that high pressure, almost everything which is there in the blood goes into this Bowman's capsule. What are those things? The plasma, that means the liquid part of the blood, all the important amino acids, glucose, salts like sodium, chloride, and all of these are kind of filtered out. What is left behind in the blood are the blood cells, which is your WBCs and your RBCs and some big proteins which are present, and platelets too. So the blood which comes out from here has very, very less plasma in it, and as a result of which it is very thick. Whereas the glomerular filtrate which gets collected in the tubule, it's absolutely dilute. Now during its journey through the tube and this network of blood vessel which is right on top of the tube, something is going to happen. What is going to happen? There is going to be tubular reabsorption. That means the substances apart from nitrogenous waste which were important for the blood are going to be reabsorbed. Like the blood would never want to lose its glucose and amino acid along with urine, right? It will also not want its plasma to go away, because that will hamper the homeostasis, or that will hamper the concentration of the blood. So water, glucose, important amino acids, salts like sodium and chloride, they are all going to be reabsorbed from the tubules by the vasa recta. and then only what is formed is called urine. Otherwise, you're not going to use the term urine, you're going to call it glomerular filtrate. Similarly, because of absorption of all of these things back into the blood, the concentration of the blood before it leaves the kidney again comes back to normal. So, all in all, what you need to understand is tubular reabsorption is very, very important, because otherwise, due to ultrafiltration, our body will lose every essential thing along with nitrogenous waste. So now we come to the next part, which is the formation of the urine. Let us see what are the steps involved in urine formation. Now there are total 3 steps in the formation of urine. The first step is ultrafiltration. Location, of course, Malpighian tubule. That means in the Bowman's capsule that contains the glomerulus. Here, under high pressure, the blood loses many of its substances into the Bowman's capsule. What are these important substances? Like plasma, like amino acid, glucose, then important ions like sodium and chloride, along with nitrogenous waste. Second step is tubular reabsorption. This mainly happens in the proximal convoluted tubule and the loop of Henle. So what all are going to be reabsorbed? Substances like water, amino acid, glucose, sodium ion and chloride ion. They are going to be reabsorbed from the PCT and loop of Henle into the blood which is present inside the vasa recta. The third step is known as tubular secretion. Again, there are certain things which are actually released before the urine is finally thrown out of the collecting duct. This happens in the distal convoluted tubule and the collecting duct. What are the substances that are added? One is potassium ions are added, and second, if you're consuming any drug, then the byproducts of the drugs, they are again released back into the urine. So what will happen after this? From the collecting duct, the urine will be sent to the ureter and then into the urinary bladder, and then from the bladder it will be released outside through urethra. This process of releasing urine from the body outside is known as micturition. So micturition is the process which is very much under control of a muscle. If you remember, in my first video I explained that there is a muscle which is present right between the urinary bladder and the urethra, and the name of the muscle is the sphincter muscle. This muscle relaxes only at the time of micturition, otherwise it always remains contracted, and this muscle only allows the passage of the blood from the urethra to from the urinary bladder to the urethra and then to outside. Let us discuss what are the various constituents that are found in the urine. Under normal condition, in the urine you will get organic substances like urea, creatinine, which is also a type of nitrogenous waste, and uric acid. Inorganic substances like sodium chloride, potassium chloride and ammonia. But there are certain medical conditions where you may find abnormal substances in the urine, like number 1, presence of blood cells. So if there is blood cells found in the urine, such a condition is known as hematuria. This condition happens if there is urinary tract infection or if there is kidney stone. Second is presence of glucose. If there is glucose in the urine, then that condition is called glycosuria. This happens when a patient is suffering from a disease called diabetes mellitus. The third condition is the presence of albumin, which is a protein in the urine. This will happen if the person has very high blood pressure or there is increased permeability of the Bowman's capsule. This happens when there is a bacterial infection prevailing in the Bowman's capsule. And the last condition is the presence of bile pigment in the urine. Well, this will happen if there is cirrhosis of liver, hepatitis or jaundice, as a result of which the urine will look extremely yellow in color. Apart from all these, let us talk about the physical appearance of urine. We all know urine is light yellowish in color. It is because of the presence of a pigment called urochrome. It is acidic in nature, and the amount of the urine that we produce is about 1 to 1.5 liters in a day. But the amount of the urine that is produced in our body, it varies, or it depends on various other environmental factors like the humidity, the temperature, the kind of physical activities that you are performing. Other than that, there is also an enzyme which controls the amount of the urine produced in the body, and the name of this enzyme is antidiuretic hormone, ADH. This is produced by the posterior lobe of our pituitary gland. Well, this hormone, when produced in sufficient amount in the body, then only we will be able to produce urine of 1 liter or 1.5 liter. But if the amount of this hormone reduces in the body, then the amount of urine produced by a patient will increase. This happens when a patient is suffering from a disease which is known as diabetes insipidus. So in diabetes insipidus, due to insufficient activity of the posterior lobe of pituitary, the amount of the hormone ADH in the body decreases, thereby causing the patient to release urine more frequently. We will study this in details when I will take up the chapter, the endocrine glands. Till then, post your doubts, keep revising and subscribe my channel. Thank you.